Hello everyone, I am Debasmita Dutta, currently working as a Ramanujan Fellow at IISER Kolkata. In this national webinar organized by ANRF and IIT Mandi, I will be briefly discussing on therapeutic mRNA vaccine project recently funded by ANRF, and at first I would like to thank ANRF and IIT Mandi for providing this wonderful opportunity to share my research project to everyone. Nowadays, whenever we talk about mRNA vaccine, it first reminds us the COVID-19 pandemic situation. However, mRNA has its own history. After its discovery in 1960s, hundreds of scientists worked for decades on mRNA vaccine before the pandemic situation happened. Vaccination strategies that were used during the COVID-19 pandemic situation by different companies and organizations. Everyone targeted spike protein as it is very unique to the virus. So if we train our immune system to induce response against spike protein, it will specifically recognize and kill the virus and virus infected cells only, but not the normal cells. Oxford and AstraZeneca as well as Johnson & Johnson, they launched DNA based vaccine that can encode the spike protein, whereas Moderna and Pfizer, they designed mRNA vaccine that can translate to spike protein, whereas Novavax, they used protein subunit of spike and it was encapsulated in the lipid nanoparticles. In case of Bharat Biotech and Sinopharm, they used the conventional inactivated whole virus vaccine. Among all these vaccination strategies, mRNA vaccine is the most widely accepted one due to its rapid production and scalability, along with the immunogenic potential. Another nucleic acid vaccine, that is DNA vaccine, has a potential risk to integrate into the host genome, whereas mRNA does not enter to the nucleus and it starts translating once it enters inside the cytoplasm. After evolutionary success in COVID-19 mRNA vaccine, we had an emerging interest to develop cancer vaccine to combat different cancer types. Now, cancer vaccine not only can act as a preventive therapy, but also it can treat the existing cancer, which is called the therapeutic cancer vaccine, and that rely on training of cytotoxic T cells to kill the cancer cells. For this project, we hypothesized that a tumor-specific antigen encoding mRNA, once packaged in FDA-approved delivery system, that is lipid nanoparticles or LNP alone or in combination with an adjuvant, that is further boost up the immune response, can selectively kill the cancer cells overexpressing that tumor antigen. As mRNA is very prone to degradation due to the abundant ribonucleases in the extracellular space, so it has to be packaged in a suitable delivery vehicle, that is LNPs. We used phospholipid, cholesterol, ionizable lipid and PEG, showing the structures at the left hand side for the preparation of the LNPs. Coming to aims and objectives, I wanna first explain the mechanism of action. The lipid nanoparticles can efficiently deliver its packaged mRNA to antigen presenting cells, that is dendritic cell. mRNA are first getting translated inside the dendritic cells and then dendritic cells present the processed antigen on its surface to educate T cells so that the T cells can further proliferate and induce tumor antigen specific immune response in microenvironment and ultimately kill cancer cells. The objective of this project is synthesis of mRNA vaccine and its characterization, followed by evaluation of in vitro and in vivo immune response and therapeutic efficacy. This work lays the framework for developing therapeutic cancer vaccine platform in India for a broad range of cancers, and patients will be benefited by therapeutic vaccination for not only the known oncogenic driver genes but also personalized neoantigen specific therapy. Our overarching goal is to determine immunogenic potential, clinical safety, feasibility and biological efficacy of therapeutic mRNA cancer vaccine and ultimately saving the patient lives. I would like to thankfully acknowledge my funding agency ANRF and my host institute IISER Kolkata to support this work. I would also like to thank IIT Mandi along with ANRF for providing this wonderful opportunity, and thank you very much everyone for your kind attention.